Good afternoon, students. Hi, my name is Ashutosh, and I'll be the TA for this problem solving session for this course, Introduction to Developmental Biology. And as you must have received the email, the link to join this meeting is will be provided in the email, and you can also contact the NPTEL team for the same. So we'll be having sessions, problem solving sessions on Saturday from 3 p.m. to 5 p.m. as you have correctly joined. So it will be a two-way session and discussion where I'll share some exemplar questions from the previous assignment, which will help you understanding the lectures better and solve your upcoming quizzes and assignments. So let's wait for 5 more minutes, and I'll keep admitting other students who are requesting to join this call. We can start. I'll share my presentation at 3:05. Till then, let's wait and see if many other students are joining. Then I'll keep them into admission into this call. Yeah. So it's already 5 minutes past 3, and I'll keep admitting students who are requesting to join this call. So I welcome you once again to this course, Introduction to Developmental Biology, and I'll be the TA for this course. And there is another of my friend who is also a TA for the same course. And what we'll be doing in these sessions is we'll solve some problem questions, practice problem set, which from the previous sessions, and which will be which will be from the syllabus of the first, corresponding to the first week of lectures. So as we progress with the practice problem session, we'll solve subsequently the second week, third week, and progressively we'll reach till the 12th week and end this course. So we'll solve some exemplary questions. I'll post the questions and we'll try to find the answers out together, and also the rationale behind and explanation behind the answers. And it will be an interactive session between the students and the TA. But for the smooth conduction of session, you guys can interact with me and my friend via the chat box. So any query or question or answers that you might want to answer, you can type your comments in the chat box and let me know. So I'll share my screen, and I'll during this session I'll please request all of the students to mute themselves and turn off their video chats. You can interact with me via the chat box. Let me share my screen now. Yeah. So students, uh, is it visible? Is my screen visible? Can you hear my voice clearly? And can you see the slide changing? Yes or no? You can please type in the chat box. Then we can begin the session. Just a minute. I'll keep admitting students meanwhile who are requesting to join the call. Yeah. So I welcome you everyone again to the Introduction to Developmental Biology course. The instructor is Professor Subramaniam. from IIT Madras. The course is offered by Professor Subramaniam from IIT Madras. He was a professor in IIT Kanpur back then. And my name is Ashutosh Behra and I'm a PhD scholar at IIT Kanpur. I hope you can see the screen. Is the screen visible? Because I think there is some problem. Students, can you just confirm again if the screen visible? I might have to reshare my screen. Thanks, Dipanvita. I will just end the screen sharing and reshare my screen again. Yeah. Let me reshare my screen. Hello. Hello. Is it visible now? Students, is it visible now? Is my audio clear? Yeah, sorry for this inconvenience. I'll try presenting my screen again and confirm with you guys. Yeah, so students, now is my audio clear and can you see the slides? Can you see the slides changing? If yes, please confirm. You can mention in the chat box. Yes or no? Yeah, great, great. So my name is Ashutosh and I'm a PhD scholar at IIT Kanpur. What we'll be going through today is practice assignment for lectures corresponding to week 1. And this course is offered by Professor Subramanian. So the way which we will proceed through these sessions is that I'll pose a multiple choice question, similar format to what you guys face in the quiz and during the assignments. Similar format of multiple choice question where one or multiple answers may be correct. If you know the pattern, professor does the exam and we'll try to find out what is the correct answer and then I'll explain about why the correct answer is the correct answer. We'll understand the rationale behind the correct answer. So it will be an interactive session and I expect students to participate and mention whatever answer you think is correct. It's okay if you are correct, if it's okay if you are even if you are wrong, you just have the courage and post your answers in the comment box. You can just type the options what you think is the correct answer. Moving on with the first question, I'll read it out loud for you guys. Which of the following statements about frog development are true? So this is the question about frog development. Which of the following statements about frog development are true? The volume of egg increases during cleavage. Option A. Option B, gametogenesis and fertilization events are seasonal events. Option C, pigment producing cells come from the endoderm. And option D, gastrulation precedes the formation of blastopore. So these are the four options. And the question is, which of the following statements is true about frog development? If you guys remember the picture of frog development from zygote to fertilization and gastrulation. So which of the one option do you think is the correct answer, students? Which of the following statement is true? The volume of egg will increase during cleavage. Gametogenesis and fertilization are seasonal. Pigment producing cells come from the endoderm or gastrulation precedes formation of blastopore. Let's take a let's take a minute or two to gather some answers. Oh, I thought you guys said that the slides are moving. Okay, let me. Now, can you see the slides are changing? Okay, sorry. I think there is some problem with connection issues. I might have to reshare. Just a minute. Yeah, now can you see the slides changing? I have changed the settings and shared my entire screen. You can let me know in the chat box, yes or no, and if my audio is clear. Sorry for the inconvenience. I think there was some connection. Is it fine now? Can you see the slides changing? Yes, yes. And students, I would request everybody to interact with me via the chat box, so that the smooth conduction of the session happens. So this was the first question that I posed. Which of the following statements about frog development are true? Now that you can see the slides, you can read the question aloud. The volume of egg increases during cleavage. Option B, gametogenesis and fertilization events are seasonal. Option C, pigment producing cells come from the endoderm. And option D, gastrulation precedes formation of blastopore. Now, which one of these do you think is the correct answer? Is a statement which is true about frog development? Let's take a minute and wait for some answers. So students who want to write their answers, you can just mention the option in the comment box. I'll be reading the comments. You do not need to raise your hands or turn on your microphone. You can just sit down, read the question, and mention the correct answer, whatever you think according to you is correct. Then we will see what is the correct answer to this question and we'll understand the rationale behind that. So let me check the comment box meanwhile, and people can take time to answer. Let's have a minute or two for every question, and then we'll try to understand the rationale behind that. Yeah, Pranay has answered option 2. roughly 10 to 12 participants. So at least I can expect some answers. No need to feel shy, and whether you have attended the lectures, it's important to attend your lectures, but if you just think logically, you can answer. What are these? What is the statement which is correct about frog development? Some people have already answered. I can see that. Ashwin says option 2. Great. Okay, now let us see what is the correct answer. The correct answer is option B. Gametogenesis and fertilization events are seasonal. So both Pranay and Ashwin are correct. Very good. Now we'll see the rationale behind why is this the correct answer. So if you see the first option, the volume of egg increases during cleavage. So what is cleavage? Cleavage is a kind of division in which there is an increase in number of cells and nuclear mass without increasing the cytoplasmic mass. It means the volume of the egg is same. So cleavage division will happen dividing the existing volume into different cells without change in cytoplasmic mass. So cleavage is different from normal cell division in which we have two identical cells or the cytoplasmic mass also gets doubled. But in cleavage we only have the division without increase in cytoplasmic mass. This means that with each successive subdivision or the cleavage, there is roughly half the cytoplasm in each daughter cell than before that division. So therefore option A cannot be the correct answer. The volume of the egg won't increase during the cleavage. The volume of egg will remain same, and in the subsequent daughter cells after cleavage, the volume of the cell will decrease. So if we see, take the whole volume of the egg, it will remain the same, but in daughter cells, we'll have the volume decreased. So cleavage never increases the volume of the egg. Now the other two options, pigment producing cells, which are also known as melanocytes, have the pigment melanin in them. They originate from neural crest cells. So there are special kind of cells which arise from the ectoderm during the event of neurulation or neural fold formation. So here is a representative diagram of an ectoderm, and this part is the presumptive neural ectoderm. So what happens? There is a invagination, and both these sheets come together to form a tube-like structure called neural tube. And this process is called neurulation or the formation of neural ectoderm. So neural structures are derived from the ectoderm, and the point of closure of the neural tube, certain special kinds of cells called neural crest cells originate. Now these neural crest cells are the source for the pigment producing cells known as melanocytes. So pigment producing cells, the third option that we saw, pigment producing cells do not come from the endoderm, but they rather are derived from neural crest cells, and neural crest cells emerge from the neuroectoderm. So pigment producing cells indirectly come from the ectoderm, but not the endoderm. And the fourth option talks about gastrulation and blastopore lip formation. So if you see the process of gastrulation in a frog embryo, this is the blastula stage where we have uh, two different structures: the outer cell layer or the trophoblast, inner cell cavity or the blastocele, and the inner cell mass. So gastrulation begins. Gastrulation is the process of movement of cells inside, movement and rearrangement of cells inside the hollow ball of cells to form the three germ layers. Now this movement does not precede blastopore formation. In fact, blastopore is the first opening in the embryo or the point through which cells can move inside, invaginate, and rearrange themselves, thereby promoting the process of gastrulation. So the blastopore lip is the first structure which forms through which cells can migrate and rearrange themselves via the process of gastrulation to form the three germ layers of the frog embryo. So here we have the representation of blastopore lip through which cells invaginate and form the three different germ layers: ectoderm, endoderm, and the mesoderm. So blastopore lip precedes the process of gastrulation. So now we saw the volume of egg will remain same during the cleavage, but the daughter cell volume will decrease, but can never increase during the cleavage. Pigment producing cells will come from the ectoderm, from the neural crest stems derived from the neuroectoderm, and gastrulation will come after the formation of blastopore, since the blastopore is the structure through which gastrulation proceeds. So the correct answer left is gametogenesis and fertilization events are seasonal, because uh, the frog development, frog gametogenesis and fertilization, frog fertilization happens during the rainy season, and this is seasonal. It doesn't happen all the time in the year. So the correct answer is option 2, as students have correctly answered. So students, is the explanation clear? You can just type yes or no in the chat box and I'll know. So this is the analysis of the first question that we have encountered. So this will be the style of solving 10 different questions. So in whole 2-hour session, we'll solve around 10 questions, exemplar questions corresponding to all the lectures in the week 1 lecture series, so that it might help you in better understanding and in solving your own assignment and problem session. I hope it is clear to the students. Great, great. So there is a chat in the comment box. Will the exam be MCQ based? Yes, yes, students, the exam will be MCQ based. It will be the similar format in which you get your assignments, right? You just have to write the same kind of questions that we solve here and the ones that you will be doing in the assignment. So the exam will be MCQ based. Sir, where can we get these PPTs? Simon Thingy says. So I'll upload these PPTs. Do not worry. The recording of this session and the PPTs, the slides will be all referred to you. There will be a Google Drive which I'll share the link, and you will get it via the NPTEL for all these slides. And I'll upload these recordings in my YouTube channel, whose link will also be provided, or I can type it in the chat box during the end of the session. You can visit the YouTube channel and you can rewatch these problem solving session, which will help you in understanding it. So do not worry about the PPTs and the recordings of these sessions. These will be available. You just have to pay attention and interest in these problem solving sessions, which I think will be fruitful for both of us, right? Now let's move on to the next question, and I expect more and more students to participate. It's just an MCQ based question. You just have to write the answers. So the next question, I hope you can see it on my slide. Questions 2 and 3 considering following image. So both these questions will be based on this image. So here in the image, we can see an insect leg, horse leg, fish fin, dolphin flipper, insect wing, and a chicken wing. Not the one that we eat in KFC, but this is a chicken wing. So the second question follows: which of the following statements are true? Remember students, there can be multiple options correct also. So you have to read each statement and analyze and then see which is the correct answer. So the horse leg, option A, horse leg and insect leg are homologous structures. For the referral, horse and insect leg. Option B, horse leg and insect leg are analogous structures. Option C, dolphin flippers, dolphin flipper and the fish fin, bird wings, dolphin flipper and the bird wings. So if you refer this diagram, this is the dolphin flipper and the bird wing. So there is a comparison between insect leg and a horse leg, dolphin flipper and the bird wing. Dolphin flippers and bird wings are homologous structures. Dolphin flipper and bird wings are analogous structures. So simple options: comparison between horse leg and the insect leg, whether they are homologous or analogous, and dolphin flipper and bird wing, whether they are homologous or analogous. So for this, you have to understand the terminology of homology and analogy, which will come later. Let's take a minute to answer these questions. You can mention in the chat box option A, B, C, D, or 1, 2, 3, 4, whichever you think is the correct. There can be multiple statements which are correct. Okay, I can see few answers. Dipanvita is saying option 2 and 3. Pranay is answering option 2. Okay. Let's wait for a minute and collect some more answers. So students, I urge you all to participate. It should be an interactive session. You can interact with me via the chat box and we'll understand the logic behind all these answers, which will help you understand your course better and also solving your assignments. Kitty is answering option 2. Ashwin is answering option 2 and 3. Great. Now let's see what is the correct option, correct answer. Before that, I'll flash the image again. Insect leg, horse leg versus dolphin flipper and bird wing. So which of these is homologous and which of these are analogous? So the correct answer to this question is both option 2 and option 3. So Dipanvita and Ashwin have both answered correctly. Pranay and Kitty are partially correct, but dolphin flippers and bird wings are homologous structures and horse leg and insect leg are analogous structures. Now let's understand the difference between these two terms. It's very important and very basic. Homology is the resemblance of arrangement, physiology, or growth of various species of organisms. So if two things have homology in them, they resemble the arrangement, the physiology, and growth of various species of organisms. So here, according to our answer, dolphin flipper and bird wings have the similar kind of arrangement. If you look at it, this is a bird wing and this is a dolphin flipper. They have similar kind of arrangement. If you follow the color coding, 5 bones here in the anterior tip. Here we have 5 bones. 2 long bones, radius and ulna, which humans also have. Here it shares the same physiology, and a small or a single humerus bone, the shoulder bone. So we see that the arrangement, overall arrangement and physiology, these two guys share this similar kind of arrangement. A dolphin flipper and a bird wing or a chicken wing have similar kind of bone arrangement. Therefore, they are homologous structure. But now analogy, on the other hand, is a functional similarity of structure based on similarity of use and not upon common evolutionary origins. So homology, we can say that if the origin is similar or the design is similar, then it's homologous. But analogous will be when the functions are similar, just like horse leg and the insect leg. So both the horse leg and insect leg are helpful in locomotion of the organism on ground. So the insect can walk on the ground and the horse can walk on the ground using the same leg. So the leg design may be different because the horse leg and insect leg are evolutionarily very different, but the functions that they share is very common. They help in locomotion of the organism on ground. Whereas dolphin flippers and bird wings have different functions. If you see, dolphin flippers helps dolphin to swim inside water and bird wings have a totally different function of flight. But why they are homologous? Since they share the evolutionary ancestry, similar design, similar body plan, and similar physiology. That's why dolphin, flipper, and bird wings are homologous due to evolutionary ancestry and common physiology, whereas based on function, horse leg and the insect leg perform the similar kind of function. That's why they are analogous. So this is a basic difference between a homologous and an analogous structure, and we should all be very clear uh, since it can be very confusing at times. Is it clear to the students? I hope now the concept of homology and analogy is clear. You can just mention yes or no in the chat box. So keep interacting with the class and with everyone through the chat box. So we'll solve such progressive questions. We'll understand the concept behind it and clarify it so that it gets cleared for once and for all. Right? Great. Great. Cool. Let's not wait anymore and move on to the third question. So third question is also based on the previous diagram. So before reading out the question, I'll want to go back and show you the diagram again for you guys to memorize. Insect leg, horse leg, fish fin and the dolphin flipper, insect wing and the chicken wing. Right? So the question number 3, which of the following statements are true, or identify the true statements? Now, owing to the same concept, we can use this in answering this question. Option A, fish fins are analogous to dolphin flippers because these structures share a common evolutionary origin. Now you have to read these options very carefully. The whole statement needs to be correct and justifiable to identify it as a true statement. Fish fins are analogous to dolphin flippers because these structures share a common evolutionary origin. Option A. Option B. Fish fins are homologous to dolphin flippers because these structures share a common evolutionary origin. Fish fins are analogous to dolphin flippers because these structures perform similar functions but have different evolutionary origins. And the last option, fish fins are homologous to dolphin flippers because these structures are anatomically similar but have different evolutionary origins. Now let us put your understanding to test. Understanding on the basis of function and on the basis of ancestry and origin and body plan. Now which of these do you think is the correct answer for this question? You can mention in the chat box. Oh great, there are many answers I can see. Dipanvita answers option 3. Pranay, Simontini, Kitty are answering option 3. Ashwin as well. Ajay is answering option 2. Okay. Now let us see which one is the correct answer. So as many people have answered very correctly, option 3 is the correct answer. Let me read out the statement aloud. See, in the analysis of the statement, we have the answer itself. Fish fins are analogous to dolphin flippers. Fish and dolphin, they might look very similar. They are aquatic animals. But if you look at their ancestry, if you look at their evolutionary origins, they are very different. Dolphin is a mammal, which is very different from the bony fish. So fish are analogous to dolphin flippers because these structures perform similar functions but have different evolutionary origins. Just now I said that fish and dolphin are of different evolutionary origins because dolphin is a mammal, as you guys all know. But these structures, the fish fins and both the dolphin flippers, perform similar functions. Because both of them are aquatic animals, these fins and flippers are useful for locomotion inside water. But they have different evolutionary origins. That is why they are analogous structures. Fish fin is an analogous structure to the dolphin flipper. So this is the only true statement. So the first option is kind of confusing you guys, might trick you into clicking the first option. Fish fins are analogous to dolphin flippers. This is true, but they do not share a common evolutionary origin. The justification is not correct. And fish fins are never homologous to dolphin flippers since they do not share a common evolutionary origin. And anatomically similar is also true for homologous structures, which fish fins and dolphin flippers are not. Because the body plan, the plan, the body plan of the fin and the dolphin flipper is different, right? But the function that they perform is similar. They are connected via the function, that's why they're analogous and different from evolutionary origin. So if we again destructurize the definition, analogous structures, these organisms do not share a recent common ancestor. So if you argue that everything came from a single common ancestor, but the process of demarcating the definition is not very recent or not very close common ancestor. They should be a little bit far apart. But they have similar appendages that evolved to help them survive in an aquatic environment. Here is an example where the penguin wing, which is the wing, will be homologous to the bird wing because they share the common kind of structure. But the penguin wing will be analogous to the shark and dolphin wing flipper because they share common functions. Penguin wing does not function in flight, whereas bird wing will function in flight. They have different functions, so they are homologous. But the penguin wing and the dolphin flipper are analogous because they both help in swimming in the aquatic environment. And here is a phylogenetic tree showing whales, including dolphins and non-whales, are mammals, and mammals diverge way before cartilaginous fish, which are sharks and bony fishes. So bony fishes and dolphins do not share a recent common ancestor. Dolphin is a mammal which is very different from a fish. They do not share close common ancestor, but the dolphin flipper and the fish fin, both help in swimming. So they share the function. That's why they're analogous. If they share the evolutionary ancestry, they are homologous, but differ in function. That's what. I hope it is clear to the students this most confusing concept of homology and analogy and the way we have to approach it. I hope it is clear to the students and students will not be further confused. If yes, just let me know in the chat box. Okay, Ajay has a question. What if origin as well as function is same? Then we classify them into the same classification category, right? Is there any example to this? Ajay, if both the origin, the ancestor, and the function is same, we classify them as the same category. Let's say, for an example, primates. We call primates because primates have both chimpanzees, monkeys, and humans. So this is a common classification since the ancestry is quite related and the function is also quite related. Primates are land dwelling. They show somewhat bipedal mode of locomotion. They have enlarged brains. So if they share common ancestry and they share common function, they are put together in a similar class. Homologous structures and analogy structures are comparison of far away. Homology is common ancestry and analogy is the functional similarity. Okay, let's move on to the fourth question. This is again a fill in the blank question with four different blanks to fill. Choose the correct option to fill in the blanks. We need to look at the dash stage for accurate classification of organisms. Alterations in dash generate dash on which dash can work. It's a lengthy question. You guys have to read it carefully. Option A, adult development, natural selection and variation. Option B, embryonic development, variation, natural selection. Option C, adult development, variation, natural selection. And option D, embryonic development, natural selection and variation. So we have four different terms here, 4 to 5 different terms, and which will fit the description. I'll wait for a moment to generate, let us collect some answers. Meanwhile, there's a confusion. I think Ajay needs a clarification. Will it be homologous or analogous? What if origin as well as function is same? If the origin as well as function is same, those structures will be called homologous organs. Homology means ancestrally they are same. So defies that rules out every other possibility. But only in a condition two organs can be analogous if they share the similar function. So the answer to you, Ajay, will be that those structures which have origin as well as function conserved will be homologous, will be still homologous organs. Now let us, many people have answered the question. Number 4, all unanimously say option 2, which is we need to look at the embryonic stage for accurate classification of organisms. Alterations in development generate variation on which natural selection can work. Sounds pretty good to me. And let us see what is the correct answer. Oh, you guys are really, really good at this. Option 2 is the correct answer. Embryonic stage for accurate classification of organisms. Alterations in the development generate variation on which natural selection can work. Now let us move on to the explanation of this answer. So if we see here, these are representative embryos for fish, amphibians, reptiles, birds and humans. And classification is possible only when we compare the embryonic stages, and differences in characteristics found in the embryo can help us classify it as a fish, amphibian, reptile, bird or a human, even if the embryo looks very, very similar in the early stages. Animals are primarily classified according to the morphological and developmental characteristics such as the body plan, symmetry, germ layer organization, and coelom are some features which form the basis of classification, which can only be found when we study, when we compare the embryos of different organisms. This is also known as comparative embryology, right? So we need to look at the embryonic stage for accurate classification of organisms. So at the embryo level, the organisms will be different. They might look similar, but they are different. They have different characteristic structures, which will classify them into different animal kingdoms or organism kingdoms. Then, alterations in development generate variation on which natural selection can work. Now, let us see how this works. Natural selection is a mechanism of evolution, and natural selection is a Darwin's theory of natural selection says that survival of the fittest. So whatever is the fittest in the current environment will survive and will yield progeny which can then transmit into the next generation. Organisms that are more adapted to their environment are more likely to survive and pass on the genes that aided their success. So this is what I was trying to explain. Whichever organism is doing well in a particular environment will pass on the genes which is helping it to achieve success in the current environment. For an example, let's say a simple example. If a bacteria present in a uh, high temperature hydrothermal vent in the sea bottom. In the sea bottom, where there are volcanic inlets and eruptions near the hydrothermal vents, very high temperature. Some bacteria can survive in the hydrothermal vent. But how did they do that? So there occurred some bacteria which have their which have special proteins which can operate in high temperatures near the hydrothermal vent. So such genes which are encoded for proteins, they are passed on to the next generation, passed on, and that's how only the bacteria now can confer an advantage in that adverse environmental condition and survive. So that bacteria is naturally selected to survive in the hydrothermal vent. And this kind of process causes species to change and diverse over time. That's how a normal bacteria and a hydrothermal vent bacteria will differ if now we compare them, because accumulation of such genes which enable its survivability in that environment has led to natural selection and caused the species to change and diverge over time. Here is another beautiful representation of natural selection and how develop variation in the development brings on natural selection. So that was what our answer told us. Alterations in development can cause variation which brings on natural selection. So here is an example of two butterflies, yellow and brown. There is a genetic variation between them within a population which can be inherited. Now, genetic variation in the genes can be inherited. So this will produce brown progeny and this will produce yellow progeny. And here is the natural selection factor. The predatory bird which will feed on the caterpillar is the natural selection factor. Now what we see is more birds can recognize these colorful yellow caterpillars. That leads to more feeding of these caterpillars and higher survivability of the brown caterpillars. The birds somehow prefer the yellow caterpillars over the brown caterpillars. That's how the brown genetic variation is passed on to the next generation. And now we can see there is an increase in population of the brown variant. Over many generations, there is a change in the allele frequency, and now this has become the gene which confers advantage to the butterfly in this environment. Just like those heat stable proteins were conferring advantage to the bacteria and the hydrothermal membrane. So in this way, an alteration in the development can cause a variation which can aid in natural selection of the organism. I hope it is clear to the students with this simple example, and you guys have answered very correctly. Option 2 is the correct answer. Is it clear to the students? You can just mention yes or no. The process of environmental factors, the surroundings and natural selection, how variation is passed on, and how there are changes in development which can get selected via natural selection. Is it clear to the students? I think we should move to the next question. People will respond. Question number 5 now. Modifying the plane of cell division inside the embryo to observe the effect on development is a dash approach. Option A, experimental approach. Option B, anatomical approach. Option C, genetic approach. Or option D, evolutionary approach. Modifying the plane of cell division inside the embryo to observe the effect of a development. What kind of approach would this be? Oh, great! Students are very quick and nimble in the responses. Good to see that. Ajay, Pranay, Dipanvita, and Kitty are answering option 1. Modifying the plane of cell division inside the embryo to observe the effect of a development is an experimental approach. And you guys are correct, very correct. This is the correct answer. So modifying the plane of cell division is an experimental approach. So here is another uh, representation of an experimental approach to developmental biology. This uh, is a transplantation approach in which the some part of the embryo or the blastopore lip of the embryo is transplanted into a new region, and here we can see the formation of secondary axis. So these kind of approaches to study embryology and developmental biology are called experimental approaches. Experimental embryology is a study of process which take place for fertilized egg to develop into an adult. So what all processes actually give rise to from a fertilized egg to develop into an adult, and with the help of physical manipulation and techniques like transplantation, surgery, changing the plane of cell division with the help of physical manipulation include experimental approach. So we do experiments on the embryo itself, on the fertilized egg itself, physical manipulations, changing the tissues, changing the cell division, transplanting a part of tissue to the other uh, part of the fresh embryo is all experimental techniques which we use to study experimental embryology or developmental biology. While there are other approaches in which we can study better the experimental embryology, sorry, developmental biology. The first strand is comparative embryology. So we had just a glimpse of comparative embryology in the earlier question when we were solving. So the embryos of fish, adult, sorry, fish, reptiles, humans all look very similar. So we compare the embryos, even if they look very similar, they have characteristic features. Some features are conserved, some features evolved and have different. The study of how anatomy changes during the development of different organism. For instance, a comparative embryologist may study which tissues from the nervous system in the fly or in the frog. So comparative embryology, which tissues, here is a perfect example, which tissues form the nervous system in a fly and in the frog. Are they the similar kind of tissue? Are they different? If they're different, how are they different? So we compare different embryonic forms of the fly, of the frog, of humans and reptiles, and then we see, try to approach that, okay, this is the similar kind of uh, body plan or some signaling event happening which is conserved and which is giving rise to similar kind of function. The second strand based on the first is evolutionary embryology, the study of how changes in development may cause evolutionary changes and how an organism's ancestry may constrain the type of changes that are possible. So we saw how evolution and natural selection is led by development and variation. So by studying the embryo and the ancestry of the embryo, so what are the restrictions the structural changes in the embryo can undergo and how development may cause evolutionary changes in an embryo. While the third approach is an anatomical approach to developmental biology, where we study the birth defects such as nasal cleft defect. Here I have shown a representation where this child has a developmental defect in which his nasal cleft hasn't formed properly. These anatomical abnormalities may be caused by mutant genes or by substances in the environment that interfere with the development. So first we study the birth defects or any defect in the organism, and we trace it back to the genes that have been mutated or have been changed which cause the organism to such. So this is another approach of studying the developmental biology. But the answer to our question is experimental embryology, in which we physically manipulate the embryo we and try to see what changes it is causing in the developmental body plan and the development of a embryo into an adult. So when we make physical manipulations, it's experimental embryology. When we compare different embryos, it's comparative embryology. When we study birth defects, it's anatomical embryology or teratology. I hope this is clear for the students. These are different forms or different approaches of studying embryology. We compare different embryos, we physically manipulate them, or we study the birth defects and the causatives of birth defects. Thereby, we can understand the whole body plan and the process of development of the embryo. I hope it's clear to the students. Just let me know with a yes or no. We'll move on to the next question. Okay, let's move on to the sixth question. So question number 6 is a match the following with the column A and column the Roman numerical column. So guys, you have to carefully match the correct options. Option A, FGF5, B, developmental force, Conklin, option C, sorry, the entry C. In D panel, we have Archenteron, somites, and neural crest. And on the right, you have to match the following. Kit gives rise to vertebrae, spinal cord, back muscles, future gut, follicle cycle, or Blumenbach tunicates. Now, which of these do you think is the correct matched option? A to 6, FGF5 to tunicates, developmental force to Blumenbach, Conklin to future gut, Archenteron to follicle cycle, somites to give rise to vertebrate, spinal cord, back muscles, and neural crest to kit. And similarly, there are other 4 options. Which of these do you think is the correct representation of the match the following column? It's a lengthy question. People, take your time. I'll wait enough time, 1 to 2 minutes. You can take your time and match the following options, whichever you think is 4. Ajay says 4, A4, B5, C6, D3, E2, and F1. E2 gives rise to that. Students can take some time and think about which is the correct option. In fact, you can just try putting these options into the match of the following and see which is the correct answer. That way you will approach your approach to this question will be faster. I see 2 students have answered. Ajay has answered option 4. Dipanvita is also saying option 4. Okay, now let us move on to the correct answer, most anticipated moment. And the correct answer is option 4. Well done. Ajay and Dipanvita have answered very correctly. So the correct match of the following sequence would be FGF5 to follicle cycle, developmental force to Blumenbach, Conklin to tunicates, Archenteron, future gut, somites, give rise to vertebrae, spinal cord, back muscles, neural crest, kit. So now let's see the explanation behind that. So Johann Friedrich Blumenbach is a scientist who studied how individuals within a species vary, and to explain such variations he proposed that a force operate on organisms as they develop. So Blumenbach give, give down the developmental force theory, saying that there is some force in play inside the organism which causes such variations among different species in the organism. Blumenbach explored the biodiversity of humans mainly by comparing skull anatomy and skin color. So, what did Blumenbach do? He classified human beings on the basis of their skull anatomy and skin color into different population, now as we know them as different ethnicities: Mongolian, Caucasian, Ethiopian, and American, based on the skull anatomy, how big and how the what is the shape and structure of the skull, and the skin color. And he proposed this theory of developmental force that causes such variations in a similar species in different individuals. So developmental force will match Blumenbach B to 5. Similarly, Conklin and Tunicates. Conklin is another scientist who took advantage of the pigmented blastomeres in tunicates to follow each fate of each blastomere. So here the yellow pigmentation shows marks the blastomere. Now tunicate organism have this special property in which each blastomere can be followed, can be fate mapped, and Conklin did that to. That's why we have Conklin matching tunicates, and this diagram here explains the gastrulation process in which archanteron will form the future gut. Archanteron is lined by the endoderm, and over it we have mesoderm and ectoderm. So the archanteron is the cavity which is displacing the blastocele here via while gastrulation. So the blastocele is the one empty cavity. After the cortical rotation and invagination of cells via the blastopore lip, there is a there is a second cavity formed which is called the archanteron, which forms the future gut. So archanteron will match with future gut. Somites give rise to vertebrae, spinal cord, and back muscles. Somites are those somites occur in early embryonic stages, mesodermal lineage, and they give rise to different somites have different specificity. They are numbered in somites are numbered in the organism, and remnants of somites are the vertebra, and the spinal cord can be sectioned, divided into different sections in the early embryonic stage called somites, and they give rise to all muscle and spinal cord, the mesodermal structures. Neural crest cells by the protein KIT. FGF5 also plays a role in follicle cycle or hair growth cycle. So the correct answer would be option 4, FGF5 for hair growth follicle cycle. Developmental force theory given by Blumenbach. Conklin experimented with tunicates. Archanteron for the future gut. Somites will give rise to vertebra, spinal cord, and back muscles. And neural crest for the KIT protein. I hope it is clear to people. This question was a little lengthy question, and I'm glad that you guys answered. I expect more participation, so people can. Yes, Ajay. KIT is a gene. KIT is a protein uh, which is found in neural crest cells. So KIT is a gene which instructs uh, the formation of receptor tyrosine kinases. So it is one of the major signaling events. Neural crest migration. So neural crests will form from the neural ectoderm and then migrate to different places and make different structures. So the KIT protein forms these signaling events which will help in migration of the neural crest cells. Okay, let's move on to the next question. The seventh question: germ cells develop from dash. So we have options: endoderm, ectoderm, mesoderm, none of the other options. Germ cells, which layer do they emerge from? Is it endoderm, ectoderm, mesoderm, or none of the other options? I can see many answers already. Pranay says option 4. Ajay, Depanvita, and Kitty are all saying option 4. So let's see if option 4 is the correct answer or not. And option 4 is the correct answer. So germ cells do not derive from the three germ layers: endoderm, ectoderm, and mesoderm. They have different origin. While endoderm, ectoderm, mesoderm are responsible for production of various organs of the body, they are somatic in nature, while germ line and germ cells are derived from other structures. We'll see them quickly. So here is a representation of a zygote which forms the blastula, undergoes different cleavage and division, cell divisions to form the blastula. And gastrula is a stage where we have gastrulation, movement of cells inwards, and arrangement into three germ layers: the ectoderm, mesoderm, and endoderm. The ectoderm outer layer forms epidermis of the skin, outer surface. Central nervous system: the nervous system neuroectoderm is derived from the ectoderm, and neural crystals. Pigment cell or melanocyte, which we have encountered in earlier questions. While the mesoderm or the middle layer will form dorsal notochord, bone tissue, muscles, and connective tissues: blood, bone, muscle. Endoderm, the internal layer, will form the digestive tract, inner lining of the digestive tract, the lung cells, alveolar cells, pharynx. While germ cells do not derive from the ectoderm, mesoderm, and endoderm, they are specified separately in the embryo, developing embryo. That's why they have a different lineage here. So if you see the precursor of germ cells, or the primordial germ cells, the origin of the germ cell lineage derive from a small population of epiblast cells that are set aside at the egg cylinder stage prior to gastrulation. So germ cells are set aside prior to gastrulation. Now only after gastrulation we have three layers: ectoderm, mesoderm, and endoderm. Even before the formation of three layers, germ cells are specified: primordial germ cells from the epiblast layer in the forming embryo. So here is an example which shows the location of primordial germ cells in a mouse embryo. So this is the gut, gonadal ridge, and primordial germ cells migrate from the hindgut and go and store themselves in the gonadal ridge. So primordial germ cells are specified very early, even before formation of three layers. While the three layers give rise to all somatic tissue structures, germ cells have the capacity to undergo meiosis to produce sperm and the egg, or the female gamete egg and the male gamete sperm. They can undergo meiosis, while these structures can only undergo mitosis, and they are called somatic cells. So germ cells are specified even before gastrulation and the three layers are formed, and they have different epiblastic origin, and these cells are known as primordial germ cells, from which the germ line is derived. I hope it's clear to the students. Now moving on to the next question. Again a simple match the following question. Which of the following are incorrectly matched? You guys have to look at the options. Which of the following are incorrectly matched? One or multiple answers correct. Hypertrophy is an increase in cell number. Malpighi and evidence for preformation. Holoblastic cleavage, only a portion of cytoplasm undergoes cleavage. And sharks, ovoviviparity. I'll wait for a minute and collect some answers. Is it option A, which is the incorrectly matched statement? Hypertrophy, increase in cell number, Malpighi, evidence for preformation, holoblastic cleavage, only a portion of cytoplasm undergoes cleavage, and sharks, ovoviviparity. Ajay says 1 and 3. Okay. Any other answers, people? Dipanvita is also saying 1 and 3. And let us see which is the correct answer. So Ajay and Dipanvita are both correct. Option 1 and option 3 are the incorrectly matched statements. Hypertrophy, increase in cell number, is incorrect, and holoblastic cleavage definition is also incorrect. Only a portion of cytoplasm undergoes cleavage. So let us see the explanation behind. Hypertrophy, if you refer to the diagram on the right hand side, is increase in size due to growth and expansion. So hypertrophy is not increase in cell number. The existing cells, we have normal cells here, nucleus, basement membrane. Hypertrophy will increase the cell size in growth and expansion, but the number of cells will still remain the same. While there is another condition called hyperplasia, in which growth of the cell in number happens due to extensive cell division. So increase in cell division is hyperplasia, while increase in cell size due to growth and expansion is hypertrophy. So hypertrophy 1, increase in cell number, is an incorrectly matched statement. Now holoblastic cleavage. So these are two types of cleavage, holoblastic cleavage and meroblastic cleavage. In holoblastic cleavage, complete division of the egg into blastomeres takes place. So the whole egg, the whole volume of the egg is divided. The cleavage plane goes all round underneath and meets its cleavage start point. So there is a complete division of cytoplasm. This is holoblastic cleavage. While meroblastic cleavage plane stops in between and there is unequal division of cytoplasm. One pole of cells, the vegetal and the animal pole cells here undergo meroblastic cleavage, forming the vegetal pole and the animal pole. So the lower part is a single cell while there are multiple cells on the top part. Vegetal cells have large quantities of yolk. So the yolk impedes the division, impedes the cleavage furrow to travel further. And here in holoblastic cleavage, we have complete division of the cytoplasm. Example, sea urchin eggs for holoblastic cleavage, and for frog eggs, we have meroblastic cleavage. Yes, Ajay is mentioning something very interesting in the comment section. Hypertrophic obesity is reversible, but hypoplastic is not. Yes, Ajay. Thanks for mentioning it. So what he is saying is that the number of cell division once increased, now there are number of higher number of cells. It's irreversible.
but the size of the cells if have increased, as in case of hypertrophy, can be reversible. Can be reversible, and the cells can gain their normal size back. So hypertrophy is increase in size due to growth and expansion, and holoblastic cleavage, complete division of the cytoplasm. Now ovoviviparity, the third option, the statement sharks and ovoviviparity. So we have sharks, we have different kinds of organisms. Viviparous organisms give birth to live pups. Both fertilization as well as development of the embryo takes place inside the female reproductive system. So humans are viviparous organism. They give birth to live pups, and the fertilization as well as development happens inside the female reproductive system. Ovoviviparous, oviparous. So we have oviparous where egg laying is the common phenomena, like in chickens. Chickens lay the egg, and all the development happens inside the egg and not inside the female reproductive system. And ovoviviparous, which are some sharks, mostly sharks, eggs incubate and hatch inside the mother's body, and babies are born live. So the egg is laid inside the mother's body, incubated and hatched inside the mother body, and live birth. So it's a transition between oviparous, egg laying phenomena, and the viviparous, live birth. So they have eggs laid inside the female body, it develops inside the female body, and the newborns are live pups given birth. And parthenogenesis is another way of reproduction in which females have reproduced without the need of male sperm. So we have a zygote derived from a diploid structure, and they do not, parthenogenesis means they do not need sexual uh, reproduction or male sperm to generate the offspring. Typically, leopard sharks reproduce sexually and are oviparous, but they also show parthenogenesis. So most sharks show ovoviviparity. Eggs are laid inside the female body, they develop inside there, and they and the development happens inside there, and there is live birth of the offspring. Yes, Ajay, you are very correct. Hollow means complete. Holoblastic is complete cleavage of the cytoplasm. So ovoviviparity is a bridging form of reproduction laying between egg laying oviparous and live bearing viviparous reproduction. This is what this is a phenomenon in which sharks uh, like rear their offspring. I hope it's clear. Yeah, Ajay, but parthenogenesis is a very rare phenomenon, mostly found in plants. You can you have some fruits which develop via parthenogenesis, and sexual reproduction is really important for human race to continue, right? As sexuality is not supported, cannot be supported by the human body. Cool. Facts aside, let's move on to the next question. Question number 9. It's also a complex question where you have to read and match the options. Choose the option in which following statements are correctly marked as true or false. Barnacles were originally wrongly classified as mollusks instead of crustaceans, and this could have been avoided if embryonic stages had been studied before classification. Statement 1. Specialized features appear earlier than general features of the group during development. Statement 2. Statement 3. Embryonic development of later organisms such as humans goes through the adult stages of development of less complex organisms such as fish. And the function of gill arches in humans is to support the jaws. Now, which of these do you think is correctly marked as true or false? You can see these options. Option 1, 1 is true, 2 is false. 3 and 4 are true. Option 2, only 4 statement is false and all the rest 3 are true. Option 3, all statements are false. And option 4, first statement is true and the rest statements are false. Embryonic development. Statement 3, embryonic development of later organisms such as humans goes through the adult stages of development or of less complex organisms such as fish. Now read all the options carefully and mark the answer. Ajay, Pranay and Dipanvita are saying option 1. Students, we are reaching towards the end of the problem solving session, so I would encourage everybody to participate. It doesn't take much time. You just have to read the question carefully, and then you will be able to guess the answer. Even if it's wrong, it's okay. We will get to the explanation behind the correct answer just after finding out which is the correct answer. So I encourage at least to answer these last two questions, question 9 and question 10. Then we'll end the session. Great. Sri Harini and Kitty have also answered option 1. Let us see which is the correct answer. Correct answer is option 4. The first statement is true and the rest statement are false. The correct answer is option 4. Barnacles were originally wrongly classified because the embryonic stages had not been included in classification. Specialized features appear earlier than general features of the group during development. This is an this is a rule given by von Baer. Von Baer laws. The general features of the large group of animals appear earlier in development than do the specialized features of a smaller group. So the general feature of the group of the a group of organism will appear first in embryonic development and later the specialized features towards the end. All vertebrates have similar structures during development: gill, arches, notochord, primitive kidneys. So all vertebrates share these common structures, but later on these become vestigial and specialized structures come into play, or these do not support the function that they support in all vertebrates, such as gill arches, which support breathing underwater in fishes, but in humans, gill arches have different function. We'll see shortly, which is also one of the statements. The gill arches form as pharyngeal arches during embryogenesis and lay the basis of essential structures such as jaws, thyroid gland, the larynx, and in mammals, the malleus and incus. So the inner ear bones, three important inner ear bones, the stapes, malleus, and incus. These are given by the pharyngeal slits, gill arches. If you study comparative embryology, in humans and in mammals, they form malleus, incus, and stapes. They also lay the basis of essential structures such as jaws and the thyroid gland and the larynx. So gill arches can form the jaws, the thyroid gland, and the larynx. Also, the muscles of facial expression and the muscles of the mandible and the jaw muscles. I think we can consider uh, the function of gill arches in humans is to support the jaws. Statement 4 says supporting the jaws, but the gill arches make the jaws in humans. So if you see, the skull is a single continuous structure made up of different bones, but the only movable part of the skull is the jawbone, lower jawbone, which comes from the pharyngeal arches or the gill arches. And not just the jaw, it also responsible for thyroid gland and the uh, inner ear parts present in the human. And von Baer laws says that general feature will arrive first, later on the specialized feature. And barnacles were originally wrongly classified, which is correct. Embryonic development of later organisms such as humans goes through adult stages of development of less complex organisms such as fish. So this is also false. So in earlier stages, earlier stages all organisms share similar kind of embryological structures, but later on uh, their development is different, and which gives rise to more complex and later organisms. Yeah. So barnacles, although they may look like mollusks with their shell-like covering, barnacles are actually crustaceans or belonging to the crab and shrimp family. Shrimp is an arthropod, sorry, crab family, uh, related to lobsters, crabs, and shrimp. Unlike mollusks, barnacles adopt novel organic matrices and cement proteins for shell formation and settlement, respectively, and the calcareous shells have potentially originated from the cuticle system of the crustaceans. So the uh, crustaceans have an outer chitinous covering. So the calcareous cells of barnacles have been derived from that. So barnacles may look like mollusks, but they are actually crustaceans. And comparative the embryology will only tell us to classify the barnacle, which has which had wrongly been classified as a mollusk, into the crustacean. In the larval stage, barnacles and crustaceans look identical, while in the adult stage they look more like mollusks. In the larval stage, they they share common features with the crustaceans. That's why barnacles fall into the crustacean category. Now, coming on to the last question of this problem solving session. This is also a match the following true false statement question. Among the organ tissues, germ layer pairs given below, identify the ones in which organ tissues are correctly matched to their source germ layer. So we just have to identify which organ is derived from which source germ layer, whichever is correctly matched. Question number 10. Oh, is the question not visible? Yeah, yeah, wait. Just a minute. I think this is the question. Yeah, can you see the question now? Question number 10, last question. Among the organ tissues, germ layer pairs given below, identify the ones in which organ tissues are correctly matched
to the source germ layer. Brain is derived from the ectoderm, heart, mesoderm, kidney, ectoderm, dermis, ectoderm, lung, endoderm, epidermis, ectoderm. So which of these are correctly matched? I think there are some answers. Option 2. People are saying option 2. Brain to ectoderm, heart to mesoderm, lung, endoderm, and epidermis, ectoderm. Let us see if it is the correct answer or not. Option 2 is the correct answer. And yes, Dipanvita, you are correct. Kidney comes from the mesoderm. So option 2 is the correct answer. Brain is derived from the ectoderm, from the neural ectoderm. Heart from the mesodermal cells. Lung, endoderm, and epidermis from the ectoderm. Yeah, Jay, you are correct. Dermis is from the mesoderm. So here is a quick revision of different cell types and layers formed from the three major germ layers. Lung cells are endodermal in origin, as well as thyroid cells and digestive cells. All muscle cells and blood cells, connective tissues are mesodermal in origin. Cardiac muscle cells, skeletal muscle cells, tubule cells of kidney is mesodermal origin. Red blood cells and smooth muscle cells are all from the mesodermal origin. And ectodermal gives rise to the epidermis of the skin and all the neural structures, neurons of the brain, pigment cells, melanocytes, with the neural crest cells, they arise from the neural crest cells. So these are different types of organs, structures, and the cell types derived from the three different germ layers. So the correct answer would be brain ectoderm, heart mesoderm, lung from the endoderm and epidermis ectoderm. I hope it's clear to the students. Uh, Someone was asking. So with this, we come to the end of the problem practice session, week 1. Let me stop my presentation. Yeah, so we have come to the end of the practice problem set week 1, introduction to developmental biology. And the coming week we'll be solving practice problem set week 2. So these are all exemplar questions for helping you understand the subject better and solve your own assignments. I hope I could help you clarify some of your doubts. If you have any doubts, you can uh, post it in the chat box, or if I'm unable to answer it right now, I'll try to solve it in the next class. I'll upload this PPT as well as the recording of this whole session. The PPTs will be uploaded in a Google Drive, and the link would be sent to you. So Ajay says question 9, option 3. I think the option said the later organisms, embryonic development of later organism follows uh,
all the stages of the earlier organisms, right? I need to present. Just give me a minute. I think this is the question which some people have confusion in. The third option, okay, embryonic development of later organisms such as humans goes through the adult stages of development of less complex organisms such as fish. So if you see the adult stages of development, the fish is totally different from the humans, right? Because but if you see the basic body plan, the vertebral column, the organs, but the fish does not have a dorsal and a ventral side, which humans have. Humans have dorsal side, the ventral side, anterior and posterior part. So at some point earlier in the embryonic development, the later organisms or higher organisms take a different course of development from where they diverge. So if you follow von Bayer's laws for organisms sharing the common ancestry, the earlier stages of development are conserved. That's why the earlier stages of embryos appear similar. But embryonic development of later organisms, higher organisms, does not go through all the developmental process of the fish. So the human doesn't become a fish totally first and then change into something else. So at a point, the embryos start developing different characteristic structures for the fish and all. So I think this diagram would be better for you to give an explanation. See, if you could see here, this is the comparative embryology. For a fish and a human, in between we have amphibian, reptile and birds. The earlier stages of embryos look somewhat similar, but the developmental process they have is very different. The human doesn't undergo all the developmental processes of the fish first and then transition into the human. I hope you get my point. For the higher organism, it doesn't go through all the developmental process of the lower organism and then switches on. So this is not a repetitive event. If according to that logic, if it was a true statement, then if fish is the lowest organism, all these organisms like amphibian, reptiles and birds would have the similar kind of development till this stage, and then they start to diverge and form their own different structures. But that's not the case. So only at early stages they are conserved and then they diverge. So they have different cases of development. Yes, Dipanvita has added to the point. After a certain stage, divergence occurs. Fish and human, yeah, they do look similar, but in earlier stages. Early embryo of human more similar to adult of the fish, you're saying. I think the early embryos of all the organisms have similar appearance and many similarity in structure and the body plan, but then divergence occurs and they have characteristic features. So the adult of the fish and early human does not look very same. If I present my screen again. Yeah, yeah, yeah. Embryo of higher developed adult human will not remain the same. Yeah. So in the similar, because it is developing, right? So in the similar species, let's say human, embryo will obviously develop. So the early stage embryo of fish and human will look similar, but once they move ahead in developmental time, they look different. Just like the example that we saw of gill arches, the gill is still present. Yeah, 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 yeah. So it not only makes the jaws, but also other organs which I mentioned, like the thyroid gland and the inner ear bones. But the supporting muscles is from the facial facial muscles are from the neural crest cells. We have a group of specialized neural crest cells called cranial neural crest cells, which form the cranial muscles, cranial nerves, and all that. So they are of ectodermal origin. But gill slits will form the jaw, which is the only movable part of the skull. Yeah. So with that, we come to the end of this session, and thank you for attending. I hope we have little better attendance for the next classes. You will also having another of my friend TA taking similar kind of exemplar questions. Please try to attend that too, and it will help you in understanding the course and solving your own assignments. Hope you a good evening, and bye bye. Meet you in the next class this Saturday, next Saturday, the same time, 3 p.m. to 4 p.m. I'll be ready with the slides, and we'll try to solve practice problem set for the week 2. Thank you. I'll end the call now.